Today's lesson is going to be on the basic molecular biology research techniques that you'll probably come across in your lab session. So as an overview, we first have to talk about gel electrophoresis because that's often going to be used in many different purposes for analyzing results. So from there, we'll talk about restriction mapping going all the way through to DNA sequencing. Now, gel electrophoresis is a technique that is used to separate charged molecules based only on their size. And it does this by using an electrical field that gets pulsed through a gel that's been submerged in a buffer solution. Now, nucleic acids are an easy one because they're uniformly charged, okay? So you have a constant charge to mass ratio, so the DNA will go from the positive pole down to the negative pole, right? Because from the negative pole down to the positive pole because DNA is negatively charged. But if we want to do gel electrophoresis on, let's say, proteins which have a heterogeneous charge, you would have to coat them in what we do coat them in is a negatively charged dye so that they continue based solely on size. Okay, so the gel matrix allows for separation on size due to the pore size of the matrix, and we have two main gels that we would run. We either have a polyacrylamide matrix, and that has a greater resolution than the other one, which is called agarose. Now, polyacrylamide can get you down to the single base pair resolution, so you can tell the difference between a 25 base pair DNA and a 26 base pair DNA, where agarose is better for, when you want, 100 base pair resolution, something like that. Now, the topology will alter the rate of movement of a DNA molecule. So nucleic acids first get linearized by being cut with restriction enzymes. So a relaxed circular piece of DNA will run slower than a linear piece of DNA, and a tightly wound supercoiled DNA will run even faster than a linear DNA. Right. So to take all of that guessing game out of it, you cut them with restriction enzymes or restriction endonucleases. Okay. So first things first. What we would do, we have a gel, okay? Let's say, for example, this is an agarose gel. You would take your sample of DNA, and usually what you do is you mix it with a colored dye so you can see it running down the lane. Not only that, you can see that you actually loaded it properly into the wells, okay? So those are wells that you make in the gel so that the molecules of DNA actually run through the gel matrix instead of on top of it. You usually always load a ladder in the leftmost well, and what a ladder is, it's actually a mixture of DNAs of different sizes, but they're all known sizes, so it acts like a ruler. Okay, so for example, you would know that, say, this is 100 base pairs, this is 200. This is 300, this is 400, this is 500, and then maybe this is 1,000. Okay, so then you can see somewhere between here, if this is 100 and this is 200, it looks like our samples are maybe at, let's say, maybe at 175 base pairs. Okay. All right, so what our gel might look like is, let's say, this. Okay, here's our gel. Okay, well, 1, 2, 3, 4, 5. Well, 1, all right, it's going to have our ladder. Okay, well, 2, let's say we have something that's right around here. Well, 3, and then maybe we even have something like this. Okay, well, 3, we have something right here. Well, 4, we have something here, maybe something here, maybe something here. So, first of all, what this is right here, we call this a smear. And a smear usually consists of many different molecules, okay, with differing sizes, or many different molecules of differing superhelical nature. Okay, so to understand that, let's talk about this. So if this would be your linear DNA, okay, that's where we would expect this piece of DNA to run. Over here, this is going to be your circular, okay, DNA. It's not going to run as far down, right? And by the way, remember, this is the negative pole, this is the positive pole, and DNA is going to run down this way because DNA is negatively charged. Okay, so your uncut, okay, your uncut circular DNA will hang out the highest. All right, your linear DNA will run really where you expect it to be. So if this is a 500 base pair marker and your DNA was 475 base pairs, that runs about where it should have ran. Okay, so that's a nice one. Over here, you would have a supercoiled piece of DNA. Okay, that's this. And over here, over here, this is an even more supercoiled DNA. Okay, the more supercoiled you are, the further you run down the gel, the faster you run. And if we had two pieces of linear DNA, okay, one that was longer, okay, so say this is a 600 base pair marker. Okay, so if this is 600 base pair, it will run in this area. Okay, and if you have another piece of linear DNA that's, say, 100 base pairs, okay, it will run in that area. Okay, so linear basically runs at a good approximation based on the ladder, whereas anything with topology, so a relaxed circle or supercoiled circular DNA won't run at the proper rate.